Imagine a world where drug discovery is fast and precise. At TopoGEN, we are dedicated to the discovery of new anti-topoisomerase drugs, critical tools in the fight against cancer. Our work is not just about finding new drugs, it's about understanding how they function inside cells to develop better, more effective therapeutics that save lives. Understanding a drug's mechanism of action is crucial. It allows researchers to predict potential side effects, off-target adverse events, and optimize therapeutic benefits. TopoGEN's insights are paving the way for safer and more effective treatments. Topoisomerase targeting drugs generally fall into two categories. Interfacial poisons, IFPs. These drugs trap the enzyme in a DNA-bound state, leading to DNA damage and cell death. Catalytic inhibitory compounds, CICs. These block enzymatic activity without forming stable DNA drug complexes, offering a different therapeutic approach. Our high-precision screening assays can differentiate between these two modes of action, providing critical insights into drug function. One of our key methodologies is the KDNA decatenation assay, which allows us to easily detect and quantify catalytic inhibitors, CICs, by assessing their ability to prevent Top2-mediated activity. This technique helps distinguish weak inhibitors from strong candidates. Here, we present data from a Top2 CIC analysis, showcasing how our platform can identify even very weak catalytic inhibition. The data show a decatenation assay in the presence of increasing levels of test drug X. First, let's look at the markers and the control lanes in the gel. Lane 1 shows that the KDNA is a high molecular weight complex that cannot enter the gel. Next lane is a linearized KDNA marker. Fully decatenated KDNA is next. The two bands are seen in this lane. The upper is Nick's circular DNA and the lower is circular DNA devoid of Nick's. Next lane shows a VP16 drug control result. The red circle is linear KDNA, which forms because of IFP activity by this type 2 enzyme in the presence of the drug E-toposide. Looking at the drug X titration from 0 to 500 micromolar, it is clear that the yield of the two decatenation products is reduced. This result shows that Top2 is being catalytically inhibited. Since we did not see any linear KDNA formation, we can assume that drug X is not a poison or IFP agent, but more likely a rather weak inhibitor or CIC drug. KDNA decatenation assays are extremely informative and help distinguish whether a drug is a CIC or IFP or both in what we call mixed mode. The assays help sort out how drugs interact with DNA and Top2, providing clarity on their effects. This knowledge is invaluable in the drug discovery process. By leveraging these assays, researchers can decode complex drug interactions, leading to new drug discoveries. Embrace this knowledge, and together we can find the next blockbuster anti-cancer drug.